Welcome to the video presentation for Chapter 8, CJC 390 for Techniques of Crime Scene Investigation. This one will be divided up into two different video presentations. It's a little lengthier than some of the other ones, so let's take a look at blood. This is a quote from the Bible, "Whoso sheddeth man's blood, by man shall his blood be shed." And blood shed at a crime scene is actually very helpful in an investigation. Obviously something we don't want to see, but when it happens it helps create events. So in these two video presentations we're going to talk about serology, the benefits of blood evidence, what viscosity and surface tension mean, the components of blood, the dangers associated with handling blood evidence, two ways that blood leaves the body in a general sense, blood stain and blood spatter interpretation, two determinations that must be made in order to form an opinion about a blood spatter's direction or origin, direction of travel or origin, and define the term void patterns, two ways blood spatter patterns are classified, velocity types with blood spatter, cast-off patterns, what blowback means, and patterns. We'll also talk about presumptive blood testing techniques, two techniques to use to locate blood stains, blood evidence preservation techniques and how blood types are determined. We'll talk about deoxyribonucleic acid, also known as DNA, three most common DNA examination techniques. We'll talk about what the benefits of mtDNA testing are and what code it is. Serology is using biological materials to identify an individual through chemical and biological tests. Students often mix up serology with the term toxicology, which is actually the study of poisons. So really, serology, we're dealing with biological fluids, biological material, connecting an individual through chemical and biological tests. ABL blood typing was invented in the very, very early 1900s by Carl Landsteiner, and ABL blood typing actually is used still today for a variety of reasons, obviously, for people who need surgical blood transfusions and things of that nature, but it is also helpful in science too. It is much easier to perform a blood test than it is to perform a DNA test. Leon Lattice actually simplified this. So what can we use blood evidence for? It can help us determine the sequence of events of how a crime occurred. Obviously, it can help us determine identity through blood testing, blood type testing, or DNA testing, and it also can determine if death occurred. There was actually a case in Kansas several years ago where a body was never recovered, but there was enough blood in the car, trunk of a vehicle anyway, that they were able to determine that there was no possible way that an individual could have lived absent that much loss. So, in looking at how we interpret blood evidence, we look at the characteristics of blood. Remember that blood starts out at least as liquid, so it moves and flows as gravity dictates. So, in a crime scene where we have a wood floor, for instance, we will not only look at the surface of the floor sometimes, but we'll look between the cracks because we know that blood goes where gravity takes it. Viscosity is the thickness of blood, and viscosity is a term that also applies to other liquids. You may hear it used with regard to motor oil, so the different weights and viscosity, 10W30, 10W40, and that sort of thing. It's merely the thickness measure of blood. Surface tension is a term that describes not just how blood, but any type of liquid is bound together so as it flies through the air, and we'll talk about this more when we get into blood spatter analysis, it holds that droplet of blood together. That's the surface tension, and also you'll see it bubble when it hits a surface. So if you can imagine a bubble of water on a countertop, if you poke that bubble of water, it breaks, and you have effectively broken the surface tension of that water. Now, the difference between other liquids, blood is a living liquid, so it has different parts to it that we'll look at in greater detail coming up. Blood also thickens over time. It tends to clot, and it separates into yellowish serum and the clot, which hardens. So we can find blood in various forms. Red blood cells, white blood cells, and platelets are the heavy phase, and we don't need to memorize these for testing purposes or anything in that matter. But just looking at the components of blood, the light liquid phase, is also known as plasma and contains serums and proteins. Blood clotting typically takes 3 to 15 minutes. Blood clotting is obviously on purpose. That's what helps seal up wounds once you are cut for whatever reason. But it's very individualized. It depends upon the viscosity in the person's blood. If blood has clotted and we find it at a scene in a clotted form, most likely it has been there for quite some time, several hours. Blood is many pathogens are passed along through blood, so we have to use universal precautions, which we referred to earlier in the course, using barrier protections, hand washing, using gloves, and things of that nature. Some of the dangers include hepatitis or the spread of tuberculosis or TB, and obviously HIV and AIDS can be passed through blood. So some of the universal precaution techniques used: avoid injuries. If a crime scene investigator cuts themselves at a crime scene, we'll typically remove them so they can be cared for and use somebody else. The different ways that blood leaves the body. So one way is if you cut yourself and you look down and you just merely see blood falling off your fingers or something like that. That is a passive way, and that's why there's a picture of this knife blade here. You can see it merely dropping, and when it drops to the floor, it creates these circular patterns because the main force that is pulling that blood or putting it in a certain direction is gravity. So passive blood tends to pool. Again, it just usually drips or runs off a larger object, which could be merely a human body, or it could be an instrument like a bat or a knife or something like that, and it forms blood. Another way is through a projected means. So there's some other force directing that blood as opposed to merely gravity. And you can see projected stains are quite different looking. The arterial spatter occurs when arteries are cut or severed. The heart continues to pump and continues to put force behind that blood to pass it through the body. And as it does so, it merely squirts out as opposed to squirting through the body. Or cast off is another way. You can see cast off is merely, and probably getting ahead of myself here, but if you get blood on an object, again, like a tire iron or something, and someone is waving that iron in a violent fashion to continue blows, blood will fly off of that object in a manner that has more force behind it, creating a pattern, as seen here in what's called. So the shape and distribution of blood, as we've already kind of hinted to, can assist in incident reconstruction so we can tell, is this a passive or a projected pattern? What are we dealing with? Blood, or any liquid, again, or any object to be honest, picks up speed until it reaches a terminal velocity. So the air resists that droppage and the gravity pulls that droppage, and the two forces eventually equalize until you reach a maximum velocity and it goes no faster. So if you were to toss something off the Empire State Building at the top level, it will not continue to increase in speed until it gets to the bottom. At some point it's going to reach its maximum velocity and go no faster. Passive patterns, again, merely dropping them down. You can see on the very right side of this picture at a 90-degree angle or straight downward creates a circular pattern because there is no other projection or velocity to the right or to the left, or however you want to say it. It just merely drops straight down. But you can see as you get more of a force behind it, or more of an angle, you can see that it creates more of an oblong or elliptical shape to it. The smaller the angle, the stranger the direction of travel, I guess you'd say, or the more um, extreme the direction of travel. So you can see 50 degrees and the difference between that and 10 degrees on the left. You can see there's quite different directions of travel. So the ones on the top of this picture here are at a different angle than the ones at the bottom. And you can also see that the direction of travel is indicated by the pointed tail end, and it actually points away. So what I'm saying is the larger part of the stain is where the direction of travel came from. You see where the arrow points downward, and the smaller tail points away from that direction of travel. The reason is that's how surface tension works when it breaks. When that bubble hits the ground, it breaks in a fashion that it sends satellite stains, what we call satellite stains, or the tail, that are smaller than the larger stain itself. So the surface tension is broken and it actually explodes, so to speak, and the tail extends beyond the original stain. So the ones at the top came first and the ones at the bottom came after. So you can see on concrete, if a passive stain in the bottom right corner on this picture, the passive stain hit concrete and it really broke the surface tension in a jagged fashion, because if you look at concrete, it's very jagged itself. It's not a smooth surface at all. So the surface can change how your stain will present too, and you can see the passive, almost passive, it's got a little bit of oblong shape to it on glass in the bottom left. You can see that it's perfectly circular almost. So this is a very good slide picture that shows how the surface tension breaks and the resulting stain, and how it's oblong when it's going at a projected angle. So you can interpret these projected patterns through multiple stains, which kind of point to a particular source, and we'll see a better slide to demonstrate that too. And the impact angle can be determined based upon the stain that you find, again dependent upon what the surface is that it hit. Another good slide that shows directionality, and again this one shows what happens when that surface tension breaks. So the parent stain in the top right of the picture shows you a large droplet, but the little satellite stains that project whenever that surface tension is broken. And you can see the direction angle going left to right, with the larger stain on the left and the tails pointing away from the direction of travel. And the bottom right one shows the same thing at a more extreme angle. When you have several paths, you can determine what we call a point of convergence, where tracking and pathway meet. And this is on a two-dimensional plane. So it doesn't give you height, but it gives you, for example, if someone's head was being struck in the center where the blue circle is here on this picture, you can see from the different stains that happened from the head being repeatedly struck where the individual's head may have been in relation to the floor. Now, how far off this individual was from the floor, point of convergence doesn't necessarily tell you that, because you need to determine the angles, and that's what we do by looking at the same stain size and the satellite stains and all that information. So here is a good slide that shows you point of origin, where you not only have the point of convergence indicated by the blue circle, but you have the purple circle in the slide picture indicating where the angles meet, and that shows you exactly how far off the floor, or with pretty good accuracy, above the point of convergence where the blows came from. Pretty great information that you can find merely by interpreting the blood stain patterns that exist. A little more difficult than it sounds, honestly. There's a lot of math involved, and much of this is done through computers these days, but it can be done. Just another picture, another slide that shows you the point of convergence and the point of origin based upon stains. And in a rudimentary fashion, it shows you that here too. And an actual picture that shows you how they located the approximate location. And you can see this is a wall. So we've got some pretty extreme projected patterns here, most likely from cast off. And at the bottom of the wall, you can see where about the K is located. There's actually a piece of trim on the bottom of the wall. computerized interpretation of different blood stains in this example. In addition to those two types of pattern, there's also something called a void pattern, and that is simply where we find an absence of evidence or an absence of blood where you would think there should be some. And how does that happen? Something was blocking the way, and that something was probably removed from the crime scene, creating a void. And that something could be the suspect, him or herself, the one who was inflicting the blows in those cases may have blocked the blood from presenting in a particular way. It may be on their clothing or their body or wherever. Different types of classifications of patterns. There's three different velocities, and two ways to classify these patterns. One is velocity and two is type. We'll talk about high, medium, and low velocity, and the two types: impact spatters, projection spatters, and combinations of the two. Low velocity spatters that we find are at a very low velocity, obviously less than 5 feet per second, fairly large, so the stains are fairly big, 4 millimeters or larger. They are from drops, passive drops, or even arterial projected patterns can be low velocity because the arteries aren't pumping at such an extreme rate as you might find cast off, as well as you might find through means, and we'll show the other means here in a second. Here's a good example of cast off. You can see how we have different angles based upon where the, say it is a hammer that had blood on it, starts and where it ends and starts its redirection. Medium velocity is 5 to 100 feet per second. You find the stains are much smaller, 1 to 4 millimeter. The impact is with blunt or sharp objects. Expirated blood. So this is blood that someone gets into their lungs and coughs up. That's going at a very fast pace, 5 to 100 feet per second, as you can see. Not as fast as high velocity, which we'll look at. The directionality and the impact angle can be used as well. And then we have high velocity, faster than 100 feet per second, very small, mist-like, 1 millimeter in diameter. And this is typically from bullets, from what we call blowback, and that is when a firearm is shot at close range. Blood not only goes the direction of the bullet, but actually backward toward the direction of where the shot came from. If the firearm is close enough, you may find this blowback on the shooter or on the firearm. Forward spatter is how it sounds. It's blood stains that follow the path of the bullet. So if it's a through and through shot, for example, it goes through someone's arm, you may find forward spatter behind where that individual was. Transfer patterns is simply as it sounds, when something, and you can see the handprint in the far left example, probably a knife print in the middle, and a shoe print in the right. Something gets soaked with blood and it touches an unstained object. So recall that we talked about patent fingerprints. You can kind of relate this to that too. When your hand is stained with something and it leaves a print.